8.2 looks at cell division. We're going to start looking at cell division in prokaryotic cells. And this is going to be fairly simple because the prokaryotic cells don't have all the organelles that eukaryotic cells do. So it can be a lot simpler process. And so they actually split through what's known as binary fission. In binary fission, you're going to get two offspring cells from the one cell that divides. DNA from the original cell gets copied, so the offspring is going to be identical to the parent cell. New cell membranes develop between the two copies of DNA. It grows until it doubles in size, and then it pinches inward and splits into two cells. So we can kind of see that here. We're starting off the cell wall plasma membrane. The chromosomes get doubled for the DNA. The cell grows and grows, starts to pinch in until it splits into two identical cells. If you're a Twilight fan, I'm sorry. This is still more romantic. Now, if we look at eukaryotic cells, this is going to happen in the cytoplasm and the nucleus, and there's going to be two types, mitosis and meiosis. In this section, we're going to look more at mitosis. So mitosis is going to be for asexual reproduction, where you just have one parent cell reproducing and making identical copies to that parent. For meiosis, this is going to involve sexual reproduction, where you have two parents and the offspring are going to be like both of those parents, but a new organism. So looking at simple cell division here, binary fission starts off as a cell, grows, pinches in, and splits into two new cells. The cell cycle, or we could say the cell life cycle. So once a new cell is made, it's going to grow and develop until it splits into new cells that it reproduces. So this growth and development time is called interphase, and there are three phases, G1, S, and G2. During the G1 phase, this is when it is newly formed. It's right after the new cell has been created. This is where the cell grows and matures into a new cell. In the S phase, this is where the DNA gets copied. And then the G2 phase is where it gets ready to perform cell division and create new cells. There's also a G0 or G0 phase, and this is if cells are not going to continue to divide. Maybe they're too mature, or maybe there's something wrong with that cell. It's not going to continue to reproduce. Normally, we do this in a couple days, but now we're going to continue it into 1 day. So mitosis, undivided attention. So we're going to use a little Kirby here to help us understand mitosis. The top row of pictures are actual slides of real cells during their different stages of division. And then here are more easy to see drawings of the different phases of mitosis. So prophase, metaphase, anaphase, and telophase. Prophase is the first phase. DNA shortens from the long twisted staircase and tightens up into the chromosomes. Then the nucleus and the nuclear membrane start to break down so that we can start that process of cell division. Centrosomes appear and move towards the opposite sides of the cell. So those centrosomes are going to be on each side of the cell. And so we've got our chromosomes there in the middle. And what is known as spindle fibers, or these are types of microtubules, are going to come out of that area. So you can see here, those centrosomes are starting to appear. There's some there. And then those spindle fibers are going to be grabbing basically the chromosomes. The next phase we have is the metaphase. And this is where the chromosomes start to move towards the center of the cell. And just like Kirby here, they're going to line up. And so those spindle fibers are going to attach onto those and help to pull them apart. So remember, the chromosomes basically have two halves. So they're going to pull one half towards one side of the cell and then one half towards the other. In anaphase, that's where that happens. So the chromatids start to pull those chromosomes towards the opposite ends. So half of each chromosome is going to one side or the other of the cell. And you can see that on Kirby as well. Next, we have telophase, where the spindle fibers start to disappear, and then the nucleus starts to form back, and the cells are actually starting to split up here. And so this part where they actually pinch off is called cytokinesis. So we're splitting into the two cells. Cytokinesis is different in animals and plant cells. For animal cells, they're going to pinch and divide, and this is going to be called a cleavage furrow. Where in plant cells, it's called a cell plate. So you can kind of see the whole process here with Kirby. So he starts off, the chromosomes are duplicated. They move towards the center. They get pulled apart to the opposite ends. They split in two and pinch off and become two new cells. There are a few spots along the way during the cell's growth and division where it kind of checks itself to make sure that it's not producing cells that are not healthy or have problems, because if we have cells that are reproducing and they aren't healthy cells, then that's a problem for our body. So 3 of those checkpoints, when the cell is growing, it helps regulate itself and checks to make sure everything is as it should be. When the DNA is synthesized in the G2, that's another checkpoint, and also in mitosis. If there's a problem with the cell and we start reproducing cells that are not healthy, or there's something wrong, maybe the DNA wasn't copied right, then this can result in cancer. And we could actually get uncontrolled growth of these cells that we don't want. And so here's a picture of actual lung cancer cells dividing, which is a problem because then they're going to make more and more.